Alright, today we're going to talk about leukemia, lymphoma, and immunity to cancer. Forgive my sniffling because I'm a little bit sick, but let us talk about genetically induced changes uh, that are the hallmarks of cancer, so the different types of genes involved. We're going to talk about two types of cancer, CLL and, or I should say ALL, and we're going to talk about briefly of how the immune system fights cancer. These lecture outcomes are taken from the actual lecture delivered to us. Okay. Um, so there are six main genetic changes that classify the necessary steps for a tumor or an abnormal growth to be sustainable and live in the body. So you need things like self-sufficient growth signals, insensitive to anti-growth signals. You need to avoid apoptosis, and you need to have limitless replicative potential, sustained angiogenesis, which means the creation or genesis of blood vessels to the tumor site. You need to also have tumor invasion to have cancer. Um, okay. Uh, while like this isn't necessarily like a hard and fast rule that this is the diagnostic criteria of a cancer, but it's just for us to better understand the molecular underlying mechanisms of what makes cancer cancer. So the last two parts, the angiogenesis and tissue invasion, is not relevant in immune cell cancers that we're going to talk about today, since they are already travel in the blood, in the vasculature, and they can also migrate really easily. So these two aren't necessarily part of it. I'm going to start with self-sufficient growth signals. Cells require signals that move them from a quiet or dormant state to a proliferative state. Um, you might remember from the first lecture of like the different stages of the cell cycle. Our normal phase is just to chill, but to enter the cell cycle, you need these mitogenic signals. In our body, in each cell, there are genes called proto-oncogenes. These are naturally occurring genes that are responsible for, when they activate, they are responsible for the initiation of mitosis. After it gets mutated, we call them oncogenes, which is a pathological expression of these proto-oncogenes, causing cells to remain in this proliferative state and leading to excessive replication. The mutation that occurs here is a dominant mutation, which means it only requires one of the two alleles to cause the conversion. We call this a one hit, and these mutations involve a gain in function because you're gaining the function of staying in a proliferative state. The example that we need to know of is this translocation between chromosomes 8 and 14. It looks a bit confusing at first. What is basically happening is you have a gene on 8 and a gene on 14. Because of this translocation, this MYC gene on 8 is now going to sit next to the gene on 14. This gene on 14 is a enhancer for the B cell receptors, which we know are antibodies. What does that mean? So this basically means that this enhancer, since B cell receptors or antibodies are stuff that is constantly activated, these enhancers, when the MYC is next to this enhancer, it continually activates the MYC gene. Uh, since only B cells are going to be affected, since you need to be next to the BCR gene, which is only activated in B cells, the MYC gene is only overexpressed in B cells. This means that excessive production or overexpression of MYC leading to excessive production of CMYC protein is an oncogenic factor for B cell proliferation, which leads to a condition called Burkitt's lymphoma, which is specifically this 814 CMYC translocation. Okay, now the next one is insensitivity to anti-growth signals. We have signals that inhibit the cell cycle, and impacting this obviously will cause it to be dysregulated. We call these genes that are anti-growth genes tumor suppressor genes, and a mutation to that is has to be a two hit since it's a recessive mutation, so it has to affect both alleles, and it involves a loss in function, the loss of tumor suppression. The example that we need to know is P53. It's a very common gene that we also, if you guys remember studying in the one of the first few lectures we got in year one, uh, it has a couple of functions. It's arrest the cell cycle, initiating apoptosis, which is the main one. Also activates DNA repair proteins. You should know that DNA repair protein activation is also part of tumor suppression. It falls under the category of tumor suppressor genes, DNA repair. Okay, and it's very common. More than 50% of cancers involve it. We can evade apoptosis as well. The basis of this is just that apoptosis, at the end of the day, you need a signal that involves proteins, and there's a cascade that occurs. What we see is actually a balance of pro-apoptotic execution of proteins and pro-survival protector proteins. When this balance is upset, we can obviously guess that we can have excess growth. The example here is the pro-survival BCL2 gene. Okay, from it's an 1814 translocation. If you remember in Burkitt's, it was also 14. Uh, this is because I'm not sure if the same enhancer is affected, but I know that another BCR enhancer is also key in causing this hyperproliferation. So same story, we have a BCL gene which moves from 18 next to 14, which has a BCR enhancer gene. Since BCR are B cell receptors, which are antibodies which are constantly expressed, these enhancers work over time and cause overexpression of the BCL2 as well. Because of this, since BCR is only active in B cells, this means there's excessive BCL2 in B cells, which means apoptosis is evaded for B cells, which means there's follicular lymphoma, which is specific to 1814 translocation. All right, we can also have a limitless replicative potential. What we mean by that is we're referring to telomeres. Telomeres, we probably already know that it's a repeated sequence of nucleotides of like I think four repeated over and over again. And the point of it is that it gets shorter every time we replicate a chromosome. And eventually we run out of telomeres, and then that causes a signal for us to scrap the cell. And this limits replication, but a telomerase enzyme is an enzyme that keeps producing these telomeres, which means that we no longer have a cap on replication, which means the cell's parent cell is basically immortal. Okay, so we talked about these four genetic changes that are required in we said that it's in an immune cell cancer. Now we're going to talk about some immune cell cancers. All right, so these words, I want you guys to make a key focus on coming back to first principles because once you read it properly, it becomes very simple. I also recommend Medicosis Perfectionalis, which is a YouTube channel, which has a series on this, and he goes through it really, really well. Leukemia, Leuc means white blood cells, emia means white blood cells in bloodstream. Lymphoma is a mass, the oma means mass, of the lymph nodes. That's the main difference between these two. However, you can imagine that if there's an over collection in the lymph nodes, that can cause circulation into the bloodstream. So lymphomas can lead to leukemias, and leukemias some of them can lead to lymphomas. So while they are different entities, they often interact with each other. For example, a condition called chronic CLL, which is chronic lymphocytic leukemia, can have lymph node masses. But we still call it a leukemia because that's the main pathology that's happening. Okay. Myeloma. Um, I want us to, I want you guys to be familiar with this lineage chart. So we have myeloid stem cells and lymphoid stem cells, which are the two main paths that a hematopoietic stem cell goes down. A myeloma arises from the B cell lymphocytes overexpression, causing like excess plasma cells, which is what the B cells differentiate into, which produce antibodies. Since you have a lot of the same B cell, you're going to have a lot of the same antibody, which means it's a monoclonal antibody instead of a polyclonal, same clone or one clone. This is, however, in the context of a cancer, it's called a paraprotein. It's not called a monoclonal antibody. It's basically is a monoclonal antibody, but it's called a paraprotein. Okay, so for leukemias, white blood cells in blood, there are four main types that we need to know. Okay, so we have acute and chronic. When we think of acute, we're thinking uh, shorter time frame, more aggressive. For chronic, we're thinking more mature cells are affected. Well, in acute, we're thinking more immature cells are affected. This will make sense in a second. So, this is another diagram, which is basically this diagram, but I've just have a bit more detail here. So, an acute lymphoblastic leukemia happens over, so it's a lymphoblast, so it must be from the lymphoid lineage. So we have a main cell here, we have lymphoids here, and we have lymphoblasts here. Okay, the key word that we need to know and understand is the difference between blasts and sites. Blasts are one step above sites, and by sites I'm referring to CYTES. Blasts are a little bit more immature, and that's why in acute stuff we have blastic stuff. In chronic stuff we have the cytic stuff. So ALL would occur here, where excess of lymphoblasts here. AML, acute myeloblastic leukemia. So we have myeloblasts here, which are the thing that's being replicated a lot. Chronic lymphoid leukemia, that is going to be further downstream over here, and same with chronic myelogenous leukemia, which is from the myelocytes, which is further downstream over here. Okay, it's fantastic. The actual objective of this in the lecture notes was to understand the difference between briefly of the ALL and CLL. Okay, starting off, age for in terms of what population it affects, ALL is the most common childhood leukemia, CLL is the most common adult leukemia. We have a little progression here. Newborn to 14-year-olds, ALL, we're thinking ALL. In the middle, 40 to 60 can be AML or CML, and over 60 is almost always CLL. Okay, so we have in both situations, sorry, this is not correct. It should say abnormal lymphocytes or excess lymphocytes, not lymphoblasts. So in lymphoblastic leukemia, you have abnormal lymphoblasts and excess of that. What this means is in the bone marrow itself, it would disrupt the hematopoiesis of other lineages. So while we're actually technically only affecting lymphoid lineage, we actually end up affecting the other lineages as well, leading to things like leukopenia in the myeloid region. We end up affecting thrombocytopenia, which is decreased platelets, and anemia, which is decreased red blood cells. So we affect this, this, and probably this as well. So this means, and same on this side. Well, we have more of the lymphocytes. The excess lymphocyte accumulation in the bone marrow will disrupt hematopoiesis. How does this present? So patients come in with uh, B symptoms or constitutional malignancy symptoms, which are going to be the same for both. So that's weight loss, night sweats, and fatigue. Uh, that is almost, these are questions you always ask across the board for any cancer, any suspected cancer. ALL is more acute, so they present with complaints more often than not. They have a fever because of leukopenia, because the body can't fight infections that well. We have lethargy because of anemia. They have bleeding because of thrombocytopenia. They have hepatosplenomegaly because you have excess These lymphoblasts spill into our bloodstream, and then they sort of um, sequester into a spleen, which is basically a big lymph node. And then that accumulation that causes portal hypertension, which leads to hepatic enlargement as well. Basically, our liver and spleen become big, and we have lymphadenopathy, which is our lymphs become swollen, but it's painless. On the other hand, CLL, since it's more chronic and slow growing, it is more often than not asymptomatic and incidentally caught on blood tests, but the same presentation here is also possible. This is curable, this is treatable but incurable. When I say curable, people go into remission for a long time. I think it was 90% at 10 years, but your lecturer will give you the specific details on that. Treatment varies because you might have been able to appreciate, for example, Burkitt's and follicular lymphoma that I talked about earlier. They involve different translocations, which means well, they likely have different therapies, right? So, for example, in CLL, a classic sort of therapy regime that we give is FCR or fludarabine, cyclophosphamide, and rituximab, which are these class of drugs. Okay, so the last part of this lecture, we're going to talk about the immune response to tumor cells. Basically, how do body kill cancer, right? Our dendritic cells, which is the major cell that brings stuff into and introduces it, brings the antigen into our lymph nodes, and sensitization and activation of these T cells, or specifically the CD8 cytotoxic T cells, this should be bolded and underlined because in cancer, this is like the key cell that is necessary to kill tumor cells. It migrates out and kills the tumor cells. So, how do does the dendritic cell identify cancer cells? That means that the antigens on the surface of the tumor cell need to be different to the self antigens of the body. So there are a couple of ways in which the antigen expression can be different. It either actually can be just normal self antigens that are overexpressed, or it can be mutated and self antigens, which allows a dendritic cell to identify it to be a cell that needs to be dead. You need an antigen. Okay, so once you bring in the tumor antigen, whether it be the APC or some other, uh, I mean, whether it be the dendritic cell or something else, what we need is an antigen presenting cell. So this cell needs to present this abnormal tumor antigen to its surface. How is it going to present it to its surface? It's going to use two MHC classes. That stands for major histocompatibility complex, and it holds an antigen up to the surface of the cell membrane, basically showing it to the immune system. Class 1 shows it and is specific to activation of CD8 lymphocytes or T cytotoxic T cells, and MHC class 2 is specific for CD4 T helper cells, which then activate B cells and leads to humoral immunity. T cell activation, MHC class 1 is which is what we want in tumors. Cytotoxic T cells eventually migrate to the tumor site and lyse them uh, and kill the tumor. There are a couple of ways that the tumor can evade our immune system. The tumor can secrete, the cells can secrete immunosuppressive cytokines, which not only inhibit the T cells, they can actually promote the infiltration of regulatory T cells or Tregs, which have a more systemic effect in inhibiting anti-tumor cytotoxic T cells, which promotes the lifespan of the tumor. Tumors can fail to produce the antigen of interest, so we said that we need tumor antigens to be different. But if they have, if don't have the specific antigen, or they don't have an abnormal antigen, or somehow it evades the system, it can mess with our immune system that way. It can also mess up the MHC1 expression, which means our CD8 T cells. Oh, sorry. Okay. What this refers to is MHC expression of the tumor cell itself, because the tumor cell, the antigens on the surface of the tumor cell are, um, they can be presented to the surface through MHC1, MHC class 1 as well. So if you don't have the MHC class 1 on the surface of the tumor cell, that means you don't have some antigens on the surface of the tumor cell. MHC class 1 is in all cells in the body. So while the tumor can avoid showing the antigen to the surface, if it's altered, uh, since there's no MHC class 1, natural killer cells know that that is an abnormal cell if the MHC1 is altered or absent, and it automatically lyses it. So it takes over for instead of cytotoxic T cells. And that is the end of the lecture. Thanks for watching.